This is the lecture on viruses, Unit 1 viruses. And I wanted to start out showing you my screen because I want to show you how to get to the slides that I'm using for the lecture. So if you go to the Wilson Community College website and then Moodle and then log in, I already had logged in, so it skipped that part for me. But go to our course, which is by 112, and then Unit 1 viruses, and it should be the first post under that. Okay, so before I start, I just wanted to say that make a comment about viruses in general. Obviously, right now is an important time to understand viruses. And so I changed the slides a little. Well, I didn't change the slides, I added to the slides a section on specifically coronaviruses and the coronavirus that causes COVID-19. When we get to our chapter on the respiratory system, we will talk more about COVID-19 and the actual symptoms that people have. And throughout the course, we'll be talking about it, probably through the discussion board, the open forum. Not going to be anything graded, but this is a particular time when there are lots of questions, lots of questions that can be answered, and lots of questions we don't have the answers to. But and I'm not an expert, but I have done a lot of research about it since I teach this subject, and I have children, and just being a concerned wife and mom, I tried to find out about it, so I can tell you what I know and what I understand about it. But before we can talk about any specific virus, we need to understand viruses in general. So one thing to understand about viruses is that viruses are not living things; they are acellular. That means they're not cellular. They're not cells. They are parasitic, which means they do infect a host cell. And when they infect that host cell, they can use the host replication processes to produce more virus. So they trick the host cell into making more virus particles. And viruses can infect any type of host cell. There are viruses that infect bacteria. There are viruses that infect plants and viruses that infect animals, viruses that infect fungi. So what you're looking at in the pictures on the bottom left is a picture of a virus. This is taken with an electron microscope. So we can't see a virus with a light microscope. But this is an electron microscope image of the tobacco mosaic virus, TMV. Tobacco mosaic virus. And then over on the right shows you what the tobacco mosaic virus can do to a plant, to the leaves of a plant. Now, some of you have more experience with tobacco than others. I know we live in a rural area, but some of you still may not be familiar with the tobacco leaf and what it looks like. But the picture here, just in case you were wondering, these are not tobacco leaves. These are the leaves of an orchid plant. And what that tells us is the tobacco mosaic virus can infect tobacco as well as a large number of other types of plants. Orchids, tomatoes, I don't know all the different ones, but I know this shows you kind of what it will do to the plant, what it would look like. Are viruses alive? We've already answered that question, and hopefully you immediately knew that it was no. Viruses exist in a netherworld between a living organism and a non-living entity. Living things grow, metabolize, and reproduce. Viruses can replicate, but they're dependent on their host cell to replicate. So they don't do it on their own. And they can't metabolize. In other words, they can't carry out processes like photosynthesis and cellular respiration. They can't convert molecules to energy or anything like that. And they cannot grow. When they are replicated, they're assembled into their mature form. They don't go through stages of growth and development. Um, One thing that's really interesting on this page, or the two images that you see, picture A and B, and you need to understand that picture B is with a light microscope, so we call that a light micrograph, and then the image A is taken with an electron microscope, so it's an electron micrograph. But what you're looking at in picture A is a bacteriophage. A bacteriophage is a virus. It looks a little bit like a spaceship. It's got a head region and then a tail region. I'm going to find a good picture. Here you go. This is a drawing of a bacteriophage. And so now when we go back to this one, you can kind of see that. So bacteriophage is a virus that infects bacteria. And this bacteriophage actually infects E. coli. So what you're looking at is just a little small portion of the cell membrane and cell wall of an E. coli cell. This is the E. coli cell, but you're only seeing a small portion of it. Now in picture B, you are actually looking at E. coli bacteria inside the intestines of some type of mammal, probably. So this would be the human cell that lines the intestines, epithelial cell. This is actually the nucleus of that cell here. So that's the human cell. And then the individual, this is an E. coli. So as you can see, you're not going to be able to see a virus attached to an E. coli cell through a light microscope. But this kind of helps you understand what is meant by 20 to 250 nanometers in diameter, because we really can't relate to numbers that small. But because viruses are so small, they weren't discovered until the 1930s, because that's when scientists developed the electron microscope. They initially were grouped and classified by shared morphology. That means shared structure. For example, the bacteriophage structure is complex. It has a head region and a tail region, and that tail region looks like it has little legs attached to it. Like I say, to me, it looks a little bit like a spaceship. But morphology just means the structure of the virus. Many viruses have a complex shape, and some have very simple geometric shapes, So later, they began to classify viruses by their nucleic acid. Now, viruses, unlike living cells, the nucleic acid in a virus can be DNA or RNA, and then it can also be single-stranded or double-stranded. We learned in Bio 111 that DNA is always double-stranded, and we learned that RNA is always single-stranded, but not with viruses. With viruses, you can have single-stranded DNA or RNA, or you can have double-stranded DNA or RNA. This is misleading. It should be hypotheses of the origins of viruses. There are three hypotheses. We don't really know for sure how viruses originated, but the devolution hypothesis or regressive hypothesis. Devolution kind of means de-evolution. So in other words, free living cells have already evolved, and then after free living cells evolved, the viruses evolved from the free living cells. They de-evolved because a virus is much less complex than a free living cell. So if a virus evolved from a free living cell, it would be kind of like going backwards as far as evolution. So we say that viruses de-evolved from free living cells. The escapist or progressive hypothesis says that either RNA or DNA actually escaped from a host cell. So RNA and DNA molecules that escaped from a host cell evolved into the virus. And then the self-replication hypothesis is saying that viruses evolved alongside their host cells. This process is often referred to as coevolution. So as the host cell evolved, the virus evolved, and they kind of evolved traits um, over time that made them, more able to the virus more able to infect the host cells, where it could more easily attack the host cell and infect it. All right. Viral morphology. And this is just morphology again, just means structure. So viral structure. First of all, when we talk about a virus, an individual virus particle is called a virion. So a virion always has two things. It always has a nucleic acid core, DNA or RNA, and it always has a capsid, a protein capsid outside that nucleic acid core. And that capsid is composed of proteins called capsomeres. Sometimes a virus will have an outer envelope, and that envelope will be composed of protein and phospholipid membranes derived from the host cell membranes. And some viruses may also contain enzymes and other proteins, enzymes such as DNA polymerase, RNA polymerase. There's one called reverse transcriptase. But these enzymes are going to be involved in the processes of replication, transcription, and translation, which we learned about in Bio 111. So viral genomes contain the genes for the proteins that the virus can't get from the host cell. Sometimes the virus will use the host cell RNA polymerase, for example, and sometimes it makes its own. So the genome of a virus contains genes for anything that the virus has to produce on its own that it can't get from the host cell. Now, the viral genome, the collection of genes, in humans, we have linear chromosomes, and we have 46 of them, and the chromosomes contain individual genes for certain proteins, and the genome is always double-stranded DNA. But in viruses, the genome can be single-stranded or double-stranded, DNA or RNA, and it can be linear or circular, and something else that's not on here is it can be segmented or non-segmented. So the genome can come in segments, or it can just be one long segment or circle. Basically, on this page, there's a lot of information, but it's pretty much just telling you there are DNA viruses and RNA viruses, and most of the time, the ones that are hardest to make a vaccine for, the ones that have a highest mutation rate, are the RNA viruses. Mutations in RNA viruses occur more frequently than DNA viruses. So you can probably figure out some examples of RNA viruses. They would be common viruses that cause common diseases that we don't have vaccines for. So HIV is an RNA virus, the virus that causes the common cold. Sometimes it's rhinovirus, and sometimes it can be an adenovirus. The virus that causes the flu, influenza viruses, and any of the ones that we, like I say, have a hard time making a vaccine for because they do mutate more frequently. Okay, the way a virus attaches. In fact, I'm going to stop here and start over with this part because we're about at the end of our 15 minutes.